All right, guys, let's take out your review sheet. You should have worked on this yesterday in class. Remember, tomorrow we're going to be having a test, and it's really important to study the review sheet because that's going to be more or less exactly what is on the test. All the major concepts, at least. The first question says, determine if the following substances are organic or inorganic. First up, we have this word organic. Remember, if you're looking at a chemical formula and trying to determine, hey, is this organic or inorganic? We're looking for two letters to be in that equation. The two letters are going to be carbon and hydrogen. The other thing that we want to remember is that there's kind of that big four category, the ones that we've been talking about this whole unit. Those include things like lipids, carbohydrates. It's also going to include proteins and then nucleic acids. The other example we have is something called inorganic. Remember, when we're talking about things that are inorganic, we're looking for things that have either carbon, hydrogen, or neither. So let's go down the list. First up, we have water. My advice is if you're not sure, write down the chemical compound. Chemical compound for water, H2O. Note H2O only has a hydrogen in it. There's no carbon present. Because of that, water is going to be considered inorganic. Next up, we have salts. We haven't talked a lot about salts. The only salt probably that you're familiar with is table salt. The chemical formula for table salt is NaCl. Note in that formula, there is no carbon and there is also no hydrogen. Because of that, that falls into the neither category. The neither category would make it inorganic. Next, we have proteins. Proteins, again, one of those big four categories. Since it's one of those big four categories, that is going to be organic. Following that, we have carbohydrates. Remember, carbohydrates is really just a fancy term for sugar. A common carbohydrate that we see is glucose. The chemical formula for glucose is C6H12O6. There we have both a carbon and a hydrogen included in there. Since we have both of those elements, we are going to consider that organic. Our next one is lipids. Remember, lipids is a fancy word for fats. Fats are, again, one of those big four organic compounds that we were talking about. So that one's going to be organic. And then finally, we have enzymes. Hopefully, we remember that an enzyme is just a specific type of protein. And above, we actually labeled proteins organic. So an enzyme is also going to be considered organic. Next up, we have what is the function of an enzyme? Remember, enzymes are going to be responsible for speeding up chemical reactions. So let's write that down. Check your answers as we go along. A lot of times people get in the habit of writing down digestion or they get in the habit of writing down synthesis. Let's not use those two examples, right? Those are two examples of chemical reactions. But what the enzyme actually does in that chemical reaction is that it helps to speed up the chemical reactions. It makes it happen a lot faster. Think back to the elephant's toothpaste that we looked at in that Ellen video. Hydrogen peroxide would eventually break down to water given enough time, right? Maybe a couple of years. But once you go and you added that catalyst or that enzyme to it, it actually was able to happen in a fraction of a second. So the real thing that's happening when we're talking about enzymes, we're talking about speeding up, increasing the rate of the reaction. 3 says, what organic compound are proteins made up of? Now, proteins, remember, that's a big organic class. So what they're looking for, more or less, are what are the building blocks? The building blocks for proteins are going to be amino acids. All right, next up, we have our second page. says, what is another name for an enzyme? And we've been going over this a bunch. You could either call it a catalyst, you might see organic catalyst, you might see biological catalyst. All of those mean the same exact thing. So a catalyst and an enzyme are synonyms for one another. And again, many times we're going to put either organic or biological in front of that. Number 5 says, explain how a high pH would affect enzyme activity. First, let's look at that word, pH. When we're talking about pH, we're talking about whether or not something is an acid or a base. Another way that we can think about this is kind of in that graph form that we've been looking at. So along the x-axis, we would label that pH. And whenever we're looking at pH, what we're looking for many times is going to be the best. The best is going to be the high point on the graph. We call that the optimum. This question is really asking, what happens once you go past the optimum and the pH gets too high? Remember, if the pH changes, it's getting higher and higher and higher, more and more basic, what's going to happen is that enzyme is going to denature. And that is going to cause a decrease in enzyme activity. So we have enzyme activity decreases because the enzyme denatures. And remember, denatures is really just a fancy word for changes shape. For our next part, we have to decide whether or not something is considered acidic, basic, or neutral. And if we are looking at a scale, remember, acids are from 1 to more or less 7. Neutral is going to be 7. And then bases are any value greater than 7 to 14. So 5 is going to be considered an acid. 7 is going to be considered a base. 13 is going to be considered a base. And then 7 is going to be considered neutral. So let's just make sure that we have that correct since I misspoke. A, or 5, is going to be considered an acid. B, 13, is going to be considered a base. And then 7 is going to be considered neutral. Our next page has us actually analyzing the graph. Hold on. Apparently I'm missing one question. So let's just look at number 10. Oh, just kidding, I've skipped them. All right, so going to 7, we are analyzing this graph. It says determine the optimum pH for the enzyme to the right. Remember, for optimum, whenever we're using that term optimum, we're referring to the best. What is the best pH in this case? What you want to do, you find out where your highest point is, and then you're going to go and you draw a line straight down. In this case, we would say it's probably somewhere between 6 and 8. With the line I drew, it looks slightly closer to 8, so I'm going to say 7.5. For a question like this, though, I would accept a range of answers. So don't be too concerned if you wrote down, oh, I thought it'd be exactly 7, or I thought it would be 8, because there will be a range of acceptable answers. If you wrote down 4, that would obviously be an incorrect answer. And when they ask you why, you're going to write down because it has the highest enzyme activity. How are you supposed to know that? Let's look at our y-axis. That's what the label is, right? That's the highest point on that graph. What happens to the enzymes after it reaches the optimum temperature? Again, now we're getting into this area here. Once you get into that area, The enzymes, the pH is so high, or the temperature gets so high that the enzymes start to denature. So we have enzyme activity decreases because the enzyme denatures, the enzyme changes shape, the enzyme is deactivated. And when those things happen, remember, that means that the enzyme's function has changed because shape is going to be responsible for determining function. What elements are present in all organic compounds? That's going to be carbon and hydrogen, just like we saw on that first page, or C and H. Again, a common representation of this, C6H12O6, where you have both carbon and hydrogen there. Once you have that, flip over. Number 10 is going along in the same way as this one. It says list 2 environmental factors that can affect enzyme activity. And the 2 environmental factors that we've been talking about this whole time, those are pH and temperature. And remember, we want to write temperature. Heat is not necessarily going to be a good answer. Think about internal, right? Internal, we're talking about what's happening to the temperature of a specific organism. 11, again, similar to what we just saw. It wants you to determine the optimum. Optimum means best. You find your high point on the graph. Draw a line straight down. Again, an acceptable range would probably be somewhere between 35 and 40. For me, I'm going to write down 37 degrees. Celsius units are important in this case. The other thing you want to note, they always want an explanation. Go over to your y-axis right once again, and this is going to be because this has the highest enzyme activity. Number 12, explain why certain enzymes work with one specific substrate. So we looked at a couple examples of this, right? We looked at lactase. Lactase specifically worked on lactose, the milk sugar. And then we also looked at the pineapple enzyme called bromelain. And that only, right, will work on protein. Why is that? That's because enzymes have a specific shape. Sometimes they call that a characteristic shape. And remember, that shape is going to determine what it actually is capable of doing. And the job, we call that its function. Next up, we had to fill out a chart. This is with the organic compounds. We had a quiz on this earlier. It'd probably be a good idea to review that quiz. The first organic compound in the example are lipids. Remember, lipids could also be called fats. When they're asking for the building block of this is going to be glycerol and fatty acids. A function of this is they have energy. Remember, this one is going to have 2 times as much energy as compared to carbohydrates. And the reason for that is simply because lipids have a lot of bonds. And remember, that is going to be where the energy is stored. The energy is stored in the chemical bonds. So lipids have a lot of bonds, and since they have a lot of bonds, that's going to be where the chemical energy is. Examples of this is going to be the cell membrane. In the cell membrane, there are these molecules. They're called phospholipids. We did have to go and look at a picture of those earlier. Remember, they have a circle and kind of 2 tails associated with them. And then the last thing on that chart is going to be the diagram. Yes, you will be responsible for knowing what each of these look like, with the exception of the nucleic acid. Just like on the quiz, the pictures will be similar to either what you see here or what you've seen on the quizzes, and then also what you've seen on the homework. A carbohydrate, remember a carbohydrate really just means sugar. A lot of times when we're talking about carbohydrates, we're talking about big sugars. The example, the big sugar that we use frequently is going to be starch. The building blocks of starch are going to be simple sugars. Remember, all the S's go together. Instead of calling it a simple sugar, you can also use the term monosaccharide. So monosaccharide, simple sugars, starch, all of those ones go together. If we're talking about what's a function of this is going to be for energy. Remember, a lot of times this is not stored energy like a fat would be. This is going to be energy that can be used in a short period of time. Think about pasta dinners. You have one before a big meet, before a big game. The next day, you're able to use that energy. Examples of this are going to be glucose. Glucose is a simple sugar. It's made up of just one ring. And then the other example is going to be a large sugar, and that's going to be starch. Yes, you do, again, need to know that diagram. You will not have to draw it, but you should be able to recognize that whenever we're looking for a sugar, we're looking for that ring structure. You're looking for that ring. Yes, there are other things associated with that ring, but for our purposes, really need to know carbohydrate is talking about a ring. Nucleic acid, there's not too much on this. We're going to have a whole unit on this later. If you're looking for a building block, we're talking about a nucleotide. Note they have the same exact root right there. The main function here is going to be in regards to your genes. A specific type is going to be DNA. And again, there are no diagrams of this, but in case you do happen to see it, it frequently has a shape like that. That picture represents one nucleotide. And last up, we have proteins, which have kind of been a main focus of this unit. The building blocks of proteins are going to be amino acids. Many times we think about this for growth and repair, and two common examples you'll see are going to be the enzyme and the hormone. Remember, those have really specific shapes. So it's important for you to remember that. They undergo that lock and key model. For the picture, the only other thing that I would note in order to help you remember this, remember proteins have nitrogen inside of their picture. Proteins out of the big three we've been talking about, fats, carbohydrates, and proteins. Protein is the only one that actually has nitrogen in there. So again, you're going to have to be responsible definitely for the building blocks and then definitely for the diagrams. The other things will be mentioned, but more or less in passing. If we are looking at the next question, number 14, we kind of covered this when we just talked about fats and carbohydrates. It says energy for organic compounds are stored in blank. Remember that energy is going to be inside of the chemical bonds. This type of organic compound contains the most energy. That's going to be lipids or fats. Remember, those are synonyms for one another. Once you have that, flip over. Last page, you'll notice at the bottom of this page, there are some review questions. Review questions from the last unit in order to make sure that we're kind of still know that information. Okay, number 15, the specific or characteristic shape of a protein determines its blank. That is going to be its function. Remember, we're talking about function, job, role. What does it do inside of your body? And we already know that enzymes have a super specific shape, and that specific shape determines what they do. Lock and key model. It says define the following terms. Synthesis means to build. Think about something starting small, right? Like you have a couple of Legos, you put those Legos together, now you've made a big tower. Digestion means to break down. Remember, for both of these, they need enzymes. If it wasn't for enzymes, you wouldn't be able to make energy, you wouldn't be able to break down your food. All of those things would take way too long. And because of that, we wouldn't be able to maintain homeostasis, and then we would die. The next one, number 17, says label the following diagram: enzyme, product, substrate. Remember, whenever we're looking for the enzyme, we're looking for the thing that does not change. So A, if we look at our picture, A is actually remaining the same. Since A is remaining the same, we're going to call that the enzyme. Remember, you can also call it a catalyst. B and C, those are going to be the substrate. And then finally, D is going to represent the product. Sometimes they'll say like, hey, if B and C are a simple sugar, then what's D, the product?
Pardon the interruption.
D, then, what is that going to be? Well, that's bonding two simple sugars together. If you bond two simple sugars together, remember, that's going to be starch. I saw a lot of that mistake on the quiz. It was the opposite way, but it was still a mistake. The other one that they can do is like, hey, what if instead now B and C are amino acids? Remember, the term amino acids is linked to proteins. There was a little bit of confusion with the building blocks, so just make sure you clear that up, review the chart, or also review this information here. Another thing to note is that this actually is a picture of synthesis. I know this is synthesis because I have two smaller building blocks and I'm going and I'm joining them together. And the last one, last little section is lab review. Lab review from labs we've actually done, or lab review talking about how to actually set up a controlled experiment. Number 18 is a lab we did in class. It says before pineapples are canned, this should actually be, let's add this. They are heated in order to sterilize them. Explain how the heating process affects the pineapple's enzymes. Think about this much like the graph. You've heated them up. Why is that problematic? Well, that's going to be problematic because now the enzymes denature when heated. Remember, denature means change shape. Change shape means now the function's changed. The last one, number 19 for this, is not a lab we did, but we can still apply the concepts, kind of like the lactose and lactase concept. It says amylase is an enzyme that works on the substrate starch. Explain why amylase cannot work on anything else. All this has to do with specific shape. Specific shape, only one match. And the very last part actually wants us to review, going way back to our last unit. It says, in order for an experiment to be valid, you can only test blank variables at a time. You can only test one variable, right? Only one thing at a time. You can't change multiple factors. So think about in our lactose example. We had one that had a pill, one that had no pill. In our pineapple, you might be like, what? We had 5 setups. But really, all that we were changing in each one of those setups was the type of pineapple. The amount of pineapple, right? For each one that we put in, we just put one little piece. The amount of jello we put in was the same. The type of jello we put in the same. But the one thing that we changed was the pineapple slash the enzyme inside of the pineapple. So we were just changing one factor there. Next up, it says differentiate between a control and an experimental. The control group is like the one that had no pineapple. The control group is the one that had no lactate pill. So that receives the normal treatment. So no drug. And then finally we have experimental. Receives treatment. So that would get the drug. There will also be a graph. If you're uncomfortable making a scale, remember that formula. You find the biggest number in your chart, and then you go and you divide that by the number of boxes. The number of boxes along the x-axis, and then for the second part, the number of boxes along the y-axis. All right, so that is it for review. Tonight, I am going to send home a copy, a handwritten copy of the review sheet on Remind. If you don't have Remind, you can still sign up. You also can sign up through your email to get Remind. And then also there are those optional Castle Learning questions if you want to do those as well. If you do have any questions and you have the Remind app, feel free to text me. I'll try to get back to you as soon as possible. You can also email me. That's a little bit more difficult for me to check, but I am more than willing to help you out if you are having a hard time. So remember, tonight, study, look over the review sheet. You can watch this video again. You can look at it on the Remind, or you can also do those optional Castle Learning questions.